All right, today we're going to be talking about muscle and how it's subdivided and the most basic subunit of a muscle, the sarcomere. All right, so here we got ourselves a whole muscle, like maybe your bicep or something, and we're gonna divide it. We're gonna show a little cross section of it, like that. And within this muscle, you have all these little striations. And these striations form a fascicle. And then within these fascicles are more striations. And each one of these striations forms, right here, popped up. This is a muscle fiber or a cell. Now these muscle fibers are also, believe it or not, striated, and we'll take a nice little subsection of that, something like that. Boom. And we have ourselves a myofibril. And these myofibrils are also have these little striations, and they're chunked up into units. These units are called sarcomeres, and they're striated, something like that. We're going to get more to that later. But these myofibrils are made up of three essential subunits for muscle contraction. We have actin, titin, and myosin motors. Myosin's like a motor protein that uses ATP for muscle contraction, rides on the actin, and the titin just supports it all. So, whole muscle, fascicle, muscle fiber, and the muscle fiber is made up out of myofibrils. Now, the basic subunit of a myofibril here is a sarcomere. And a sarcomere will look something like this. Wider. And so these little lines here, these are actin filaments, and subdividing each sarcomere is something called a Z line. And the actual myosin motors for these actin filaments, they sit in between each of these these are called thin filaments, and we got these thick filaments. Look like little boxes under a microscope. Okay. These are the myosin. So actin and then myosin makes up these little boxes in the middle. Now subdividing the sarcomere is a structure called the M line. And if you were to look under a sarcomere under this microscope, you would get these little bands that form, and you get these I bands and these A bands. Now an A band is this dark region right here that comprises the thick filaments. And an I band would be this lighter color region out here. Now the zone between these two thin filaments is called the H zone, what that is called. And titin will kind of sit right here and support these thick filaments. This is titin, kind of stretching out and supporting them. So that in basic, it is a sarcomere. For muscle, when muscles contract, these little myosin motors have these little heads that come up and bind to the actin. And in a mechanism that we'll discuss later, they basically move down the muscle, causing the sarcomere to kind of compress like this, and causing a muscle contraction down the whole muscle fiber itself. And so that is basically the basis, the most basic building block of a muscle, the sarcomere.
If you notice anything with the numbers, that was
It just mirrors this question we had.
Okay, let's talk about
With the formula
how calcium gets released in the sarcoplasmic reticulum of muscle fiber, and how it activates myosin motors to cause muscle contraction. Okay, let's go.
Oh yeah, you gotta do stupid. What is it
Okay,
called?
within a muscle cell,
Yeah, you have to do
there
that.
are a ton of these little indentations.
And then I think the 3 moles of hydrogen means that you divide that number by 3, and that's your rings.
Something like this. It's not exact, but something like this. And within these T tubules actually are comprised of extracellular fluid. So there's calcium ions floating around here. There's all different kinds of ions. And within these T tubules, there are these L-type calcium channels called DHP receptors.
Yep. So when you get your charges set up with anything,
so it's a little misleading that they're called L-type calcium channels because they don't actually let
I'm looking at that.
calcium
I'm confused. I know
through.
that I've learned that at some point. In the
Rather,
past,
what
but
these DHP receptors do is they act more like as like a voltage sensor,
then we have
and they're hooked up here to this another channel, which is embedded in the membrane of the sarcoplasmic reticulum. There's another little pump here that we'll discuss later.
magnesium 1.
Okay, this is the sarcoplasmic reticulum. Within here,
So when you just get the charges,
it's called the smooth ER in other cells that aren't muscle cells, but within here there's tons of calcium ions just ready to spill out into the ICF. So we get an action potential that depolarizes the membrane, and it will actually cause this DHP receptor to open.
to figure out what you need.
Kind of like click open like that. And this receptor is connected to an RYR receptor, or to an RYR channel. And this channel is mechanically gated, so when this DHP receptor opens like this, this mechanical stimulus will actually open the RYR receptor here, and it will cause calcium to spill out into the sarcoplasmic reticulum. And this calcium will cause muscle contraction, which we're going to talk about here soon. And to end a contraction, because we don't want to just keep contracting our muscles, we have the Circa pumps, which is just a calcium pump to pump the calcium back into the sarcoplasmic reticulum. Okay, all right, let's talk about how muscles contract at the molecular level. Okay, so we already talked about how in a sarcomere we have these thin filaments, and then we got these thick filaments, right? These thick filaments are the myosin motors, and these thin filaments are the actin subunits. Now we're going to zoom in here onto this little bit right here, and here we got our little myosin motor, and along these myosin motors are these heads.
So you just look first on what it begins with. So if it's a
And
metal
here's the thin
or
filaments.
NH4, then you just know. Okay,
Now,
it's an ionic
when
compound.
ATP is bound
Then is it
to these myosin heads,
predictable? So if it's not,
they
then
are
it's
in
super
their
set.
uncocked, relaxed state.
If it is predictable, then
Also,
it's
there's
atomic. If
this
not,
protein
it's
called
just
troponin C
It's just something to help you.
binding this
No, that's good.
this actin filament. Actin.
So that's something you can just look at. Even like copying it down.
Okay.
Memorizing it.
Yeah. Yeah, I want to make sure I copy this myself.
There you go.
Oh crap!
Oh, yeah,
I forgot. Okay, now that we know that calcium gets released to the sarcoplasmic reticulum to get the muscles to contract, now we're going to talk about how that happens at the molecular level. So here within our sarcomere, we already talked about how we had this myosin motor these actin filaments. The myosin itself, we zoom in, sort of has this like weird head shape, and it'll kind of like walk along the actin, causing an actin to move on it like a track, and that actually causes the muscle contraction. So bound to this myosin, right now it's in what is called a cocked state, kind of like a gun. It's just cocked back and it's ready to fire, go forward. It's bound to ADP. ADP and inorganic phosphate are bound to it. And here is our actin track above. Right now it's unbound. And each myosin binding site, there is a troponin C protein here. And it's sort of covering up the binding site. Now, calcium, what it does is when it floods the cell, will actually bind to these troponin C proteins on the actin track. And when it does so, it sort of causes the troponin to rise up and expose the myosin binding site. So I'm going to redraw it here. So it'll bind, and then it'll expose these myosin binding sites. And the troponin C will be bound here and bound here. And now these myosin heads, they will bind, they will release this inorganic phosphate, and they will bind to the actin track with ADP still bound. And so to actually get the power stroke, what will happen is this ADP will unbind, and the ADP sort of, the charges of the ADP sort of keep it cocked in that high energy state. And when the ADP unbind, the heads will move forward like this to their relaxed state, sort of bringing the track forward. And then so they move forward, and now they're in their relaxed state. Track moved forward a little bit. And now to unbind the myosin, ATP will come in and it will bind to the stem. And this releases the myosin off the track, like this. Myosin's released. And now calcium will unbind, and then the troponin will come back and recover the binding site for myosin. And now, to cock the myosin back into its cocked state, ready for another power stroke and another contraction, the ATP will actually hydrolyze here, and we'll get ADP, still bound to the, still bound to the myosin. And then inorganic phosphate from that ATP molecule. And that'll bring it back to its original cogged state, ready for another contraction.
Okay, that's good to know.
Like, get into the textbook and just take lots of notes.
Okay.
Like, do that stuff.
Yeah. That's good to know.
Okay, now that we know like how muscles contract, we're going to talk about like what powers the muscles to contract. How do they get ATP, or what types of ATP come in? So first off, we have cytosolic ATP. This is ATP just floating around in the cytosol of the cell, and this source of energy lasts for about 5 seconds under maximal effort, and it's good for quick bursts of energy under anaerobic conditions. And so when we expend our cytosolic ATP, our muscles will rely on another source of energy called creatine phosphate. And what this does is, it's a high energy molecule, and it will freely donate its phosphate away to ADP to form ATP. I'll just draw this out. So creatine phosphate and ADP. There's an enzyme that catalyzes this, will give us ATP. And this will give us about 10 seconds. And this is good for, this is another quick sort of energy source for anaerobic slash maximal intensity contractions of skeletal muscle. Third, we're going to talk about that's anaerobic. We have glycolysis. And this can also happen under anaerobic conditions. This is just the breakdown of glucose, glycogen. You can store about 200 grams of glucose in your skeletal muscles, a little bit more in your liver. And so the glycogen will break down into glucose, and then this glucose will get broken down in the cell via glycolysis, and it'll make ATP under anaerobic conditions. And this lasts about a minute under, again, anaerobic conditions where your muscles are just going hard. So that's what the muscles use for anaerobic conditions mostly. And now we're going to talk about aerobically, like say you're running, swimming, biking, hiking, any sort of cardio workout, and you're going to be using, or I guess I should just say oxidative phosphorylation. This is the electron transport chain. And this works only under aerobic conditions. And it's very slow, so it's not very good at replenishing the muscle quickly. So it's not good for sort of the maximal activity. Say you're like lifting weights, or you're lifting something heavy, or you're trying to sprint. It's not very good for that. It's a slow form of ATP synthesis. But the benefit to it is it lasts for hours. It'll last a very long time. And whereas glycolysis was mostly reliant on glucose, this one can use glucose as well. Glucose is a good source of energy, but an even better sort of source of energy for oxidative phosphorylation are fatty acids. So yeah, those are the 4 different sources of ATP for skeletal muscles. Okay, sweet. Now that we've talked about skeletal muscle, its contraction, we're going to talk about smooth muscle. Smooth muscle. Smooth muscle can be subdivided into 2 categories. We have single unit. Single unit smooth muscle is a smooth muscle that contracts via peristalsis is what it's called. It's like a wave-like property. Like one side it contracts, one side relaxes, and it kind of just moves down like a wave. I imagine like the small intestine pushing food as it digests. Single unit, cells or joined together via gap junctions. So something like this. Or that's one cell, and then there's a gap junction, and they can communicate with one another. The gap junctions move together as one cohesive unit via peristalsis. That's a single unit muscle. Now, let's talk about multi-unit. And these singular cells depend mostly on endocrine or nervous system input. So, single unit, multiple cells come together, they act as a single unit to move food, or, work as a smooth muscle and move via wave-like contraction. Whereas multi-unit, it's different, as the cells can act as different units, or like there's like a neuron that comes down to tell them to move, and they can either contract or they don't rely on other cells basically via electrical signals and gap junctions to know whether to move or not, contract or not. And that's the difference between multi-unit and single unit smooth muscle. All right, we're going to talk about smooth muscle contraction. Smooth muscle, like skeletal muscle, will contract. The difference between smooth muscle and skeletal muscle: smooth muscle is a little bit more slow. It's very strong though, and it will be able to contract for hours. It makes sense why our bowel movements, we don't really get tired, they just happen. So, we're going to talk about two different types of smooth muscle contraction: phasic and tonic. Which phasic contraction here, we'll have our same actin thin filaments and our myosin motor here. Here, it's in the contracted state. ADP is bound to it. ADP, and then we got phosphate. Okay, so to get this contraction going here, we'll have extracellular calcium flood in. Smooth muscle requires more extracellular calcium than intracellular calcium to activate, to work. And so 4 calciums will come here and they will bind calmodulin, a protein. And this calmodulin will become active. And this calmodulin will activate MLCK, activating it, something like this. Active. That myosin light chain kinase will come here and phosphorylate this motor protein. Now, it will bind, but it will have 2 phosphates on it. That's the phosphorylation step, and the ADP. Now we get our power stroke. And it will lose an ADP and a phosphate. Get our power stroke, it'll come forward. It'll be bound here. And then ATP will jump on again. It'll come off. Then it can go back to this state right here and just keep cycling through, where hydrolyzed ADP jumps back on, and it will just keep doing this. That's why smooth muscle is really good at contracting for hours, because it can do this little system here where ATP will keep jumping on and it'll keep hydrolyzing and it'll do this cycle. And this will keep going until myosin light chain phosphatase gets active here and we get relaxation. MLCP. And so that's more or less how phasic contraction works. All right, now we're going to talk about tonic smooth muscle contraction. And it's very similar to phasic here. So, there's just a couple key differences. It's like in phasic contraction, well, I guess phasic contraction is just like muscles can just contract, relax for hours due to that state where they can just be cycling through ATP. This one's really interesting. So, here in the tonic state here, we start off with our ADP, or phosphate, right? Then here's where it gets interesting. Well, actually, no, it's the same thing. So, our 4-calcium will come here, bind calmodulin, like in the last one, activate it. It'll then activate MLK, myosin light chain kinase. That'll be active. And then it'll phosphorylate just as it did last time, right? But here's where it gets interesting. So we have our, it's ready to go, ready to power stroke here. 2 phosphates and our ADP. But then here, when it power strokes, our myosin light chain phosphatase will actually come in right here. And it'll dephosphorylate it. And so we'll get we got 2 phosphates off of it, and then our ADP. And now when this happens, it's already constricted. It looks something like this. It already, contracted. It's contracted right now. It's like this. Ready to, it just stays latched. A molecule of ATP will come onto it, but it won't release. Rather, it'll just stay on there. It won't hydrolyze. It'll stay on here. In a phenomenon, what's called a latch state. And it will take forever to relax. This is called the latch state. Bridge, also latch bridge. Here, it just stays constricted, contracted for hours potentially, or indefinitely without fatiguing. And it's this phenomenon is what's really amazing about smooth muscle, the fact that it can just stay constricted, contracted like this for hours due to this. And so this is tonic smooth muscle contraction. All right, now we're going to talk about the differences between skeletal and smooth muscle. We're going to talk about functional, regulatory, and structural differences. First off, we'll talk about structural differences. So skeletal muscle striated contains sarcomeres, little sections. Smooth muscle is, well, smooth. Sarcomeres, no sarcomeres. And that way it's different. Skeletal muscle has T tubules. Smooth muscle does not have T tubules. Skeletal muscle cells, they're multinucleated. Singly. So there's just some structural differences. All right, let's talk about functional. All right, skeletal muscle, functionally, it's voluntary, involuntary, somatic autonomic nervous system, fast, slow, skeletal muscle, fatigues easily, and smooth muscle is fatigue resistant, and that it can contract for long periods of time. This is mostly meant for motor movement. And that's mostly meant for the movement of fluids, liquids, yada yada. All right, let's talk about regulatory. All right, skeletal muscle solely uses acetylcholine. Smooth muscle can be activated, inhibited, etc. by acetylcholine, epinephrine, others, and other neurotransmitters. Regulatory skeletal muscle uses troponin as its skeletal binding, as the binding protein for actin. This one uses calmodulin. Skeletal muscle has a lot of SR, calcium, and SR. This one mostly uses extracellular calcium. Skeletal muscle is innervated at motor end plate. At the motor end plate, and in smooth muscle, you'll find nerve endings along the whole tube, like the cells. And those are some differences between skeletal and smooth muscle. Okay, we're going to talk about how muscles, sort of how like reflexes work and stuff. So we got two types of muscle sensory fibers. We have muscle spindles, or spindle fibers, I should say. Spindle fibers. And we got Golgi tendon organs. So first off, we'll talk about muscle spindle fibers. So within our muscle here, we got these things called the muscle spindle apparatus. We have two types of fibers. Bag fibers. We got chain fibers. All right. Bag fibers are very sensitive to stretch. And chain fibers are there just to maintain posture. These spindle fibers are huge in just maintaining posture and our general reflexes, and they're very sensitive to stretch. Something else I should talk about too is that when these are activated, they cause a contraction to counter the muscle stretching out too much. Yeah. So Golgi tendon organs. Here's our muscle. They're kind of like right here, between the muscle, where the muscle transitions to tendon. Muscle. That's a Golgi tendon organ. And they resist tension. Once we get like too much tension, it will send an afferent signal to the control center to stimulate motor neuron to cause the muscle, or inhibit the motor neuron to cause the muscle to relax. Causes muscle to relax. So Golgi tendon organs primarily deal with tension, where spindle fibers deal with stretch. So yeah. Okay, now we're going to talk about the stretch reflex. So, you ever been at the doctor's? All right, let's again. Now we're going to talk about the stretch reflex. You ever been at the doctor and they like hit your knee, your leg kicks up? Yeah, that's the stretch reflex. So, here's what we do. It has to do with muscle spindle apparati doing their thing. Use your quad, use your hamstring, use your leg bone or something, use your leg. So, doctor will hit the kind of like this tendon here with like a hammer or something, but cause your leg to kick up. So here we got this sensory neuron connected to the muscle spindle apparatus. Muscle spindle apparatus there. Let's send a signal out to the control center. And basically what will happen is the alpha motor neuron going to the hamstring be inhibited. And the alpha motor neuron will be activated going to the quads. And that will cause contraction and movement of the leg to resist the stretch. That is as simple as can be the stretch reflex. Now let's talk about the withdrawal reflex. Have you ever like touched a hot stove? You draw away quickly. That's called the withdrawal reflex, and that's to protect us from things that can potentially harm us. Here we go. So withdrawal reflex. So throughout our body, we have receptors called nociceptors, which are sensitive to pain. So say you touch a hot stove. And you touch it, you're an idiot, and so you touch it with your hand. It'll detect pain, and it'll send a signal to the control center. And then basically extensor muscles, extensors inhibited, flexors activated. And then draw away. Ouch. That makes sense. Your extensors in, say your arm, which would be like your tricep, they want to move out. Your flexors, like your bicep, they just want to move you back in. So as soon as you touch that hot stove, you're like, ah, crap, but really fast. The nociceptor takes pain, 350 miles an hour, goes to your brain, boom, boom, activates the bicep, alpha motor neuron in the bicep, and it pulls away real fast. It's like, I don't want to get hurt. Maybe a little bit of your, like your chest, maybe your shoulder a little bit will get activated. So yeah, withdrawal reflex. Those are 3 different reflexes: stretch reflex, withdrawal reflex. This has been my video. Thank you. Good night.